Good morning, everyone. Today I'm going to teach you biotechnology principle topic, which is a very important topic as in your class 12 course. So, starting with the selectable markers, which is the most important part of it. So, what all things you need is vector, which is a plasmid in case in your code, and a target gene or the gene of interest, which you need to insert it into the vector, and through that vector will get inserted into the host cell, which in our case we are taking bacteria. So how this process take place when we grow it into the medium, or you can say petri dish. What happen is some of the bacteria does have the vector, and then some of them it does not get inserted. So how would we distinguish between both of them? So for to that we have a technique, um, which is um, which is due to the selectable marker. We call it transformant and non-transformant DNA molecule cell. Transformant cell is basically of two types, one non-recombinant vector and recombinant vector, and non-transformant bacteria is the one which does not have even the vector to be entered. So how would we distinguish between both of them? So the first step is we will take a petri dish and put a medium of ampicillin or tetracycline antibiotic in it, which the bacteria, the vectors, one the recombinant and the non-recombinant vector bacteria does have the antibiotic resistance present in it, whereas the cell of bacteria in which no vector is present, so there would be no antibiotic resistance present over there. So to resolve this problem, to separate non-transformant, to make deadly non-transformant bacteria, what we will do, we will put it into the ampicillin solution. So we will observe that the non-transformant gene DNA cells are dead. Now we have the second purpose is to separate recombinant DNA cell and non-recombinant DNA cell. So the recombinant DNA cell, we will put BAMH1, which is a restriction endonuclease, which will be entered into the site of the selectable marker. Between the selectable marker, the selectable marker will get inactivated. As the selectable marker will get inactivated, it will lose its property of resistance. Then the second cell which we will be taking is, we won't be inserting ampicillin or any kind of endonuclease resistance enzyme. So what we will observe would be, we will put it into the tetracycline solution, where we will see that the one will get dead and the one which we want will be okay.